Today we're going to be continuing on with chapter 6, exploring microbial growth and nutrition. Specifically, what are the parameters for certain microbes to grow? What pH, what temperature, what pressure do they need to survive and thrive? So some of the environmental factors that influence microbial growth can be heat or cold. So each microbe, actually each living thing, whether it's a plant or ourselves, has a certain parameter that they will scrape by in and a certain parameter that they will thrive in. There's also radiation. So this would be like UV light, sunshine. Is it helpful, hurtful? How do they respond to it? What gases are needed and which ones are tolerated. So things like CO2 and oxygen become very important when we talk about microbial growth. Also pH, so how acidic, how basic is the environment that they find themselves in. This comes up a lot when we talk about microbes associated with human health, because the microbes in the stomach, where we have a very low pH, are not going to be the same as the ones we would find on the skin, which wouldn't have that very acidic environment. Okay, so based on where you find them, you'll also see what sort of pH they prefer. We'll also talk briefly about osmotic pressure, or the pressure from the environment or the atmosphere, or from water if they're in the ocean, things like that. And also what microbes are in their environment. So we know that while we might grow a pure culture on a dish, ordinarily they're not going to be the only microbe there, and their own microbes in their biome will affect them greatly based on what compounds they excrete, whether they work together or they try to fight each other off. The other microbes play a key role in their environment. Okay, so we're going to go through each of those, starting with temperature. Microbes cannot control their own temperature. So I have a little thermometer on the left here. They don't have their own thermostat. So like ourselves, we can try to regulate our internal body temperature to be 98.7 all the time, whether it's snowing or if you're in the Bahamas. Okay, microbes don't have that luxury, and therefore they will take on the temperature of their environment. So if you put them in the fridge, they're going to be the temperature of the fridge. If you put them out in the sunshine, they're going to warm up, understandably. Okay, so the range of temperatures for the growth of a given microbe can be expressed as a cardinal temperature. Okay, so the cardinal temperature is that range that they prefer. And you usually get three numbers when you talk about cardinal range: the minimum, the maximum, and the optimum temperature. So the minimum is that cutoff where they're barely scraping by. Oh, now they're not going to be making it anymore. It's going to be on the lower end. So when does it become too cold for them to make it? Maximum, similar idea. It's going to be when does it become too hot for them to survive? And then we have the optimum temperature. This would be the small range where they're the happiest, where it's the ideal growth conditions. And it's interesting that we can use these numbers to manipulate the microbes. So if I'm trying to grow them in the lab, I'm going to want to know that optimum temperature. And if they like 37 degrees Celsius because they grow inside the human body, I want to be growing them at 37 degrees. On the other hand, if I'm trying to regulate or control growth, minimize growth, then maybe I need to know the minimum and maximum temperatures so that I can maybe make it so cold that they can't survive, or make it so hot that they can't survive. So it depends on your goals. Are you trying to get rid of or regulate their growth, or are you trying to grow it for research purposes? Different microbes will have different cardinal ranges. So for each one, depending on the species that you're studying, you'll need to go and look at, okay, what's the minimum, what's the maximum, what's their ideal? And this really depends on where you would normally find the microbe. So some parasites will grow in temperatures of only a couple degrees from their host body. So one example is like a rhinovirus we associate with the common cold, okay? It has a range of 33 to 35 degrees Celsius. So it's going to be slightly cooler than the inside of the human body at 37 degrees. So we'd probably find it on the outside, maybe in your mucosal membranes, like where you'd be sneezing and things like that. That one has a very small range, so it's only going to survive very well in that tiny window. On the other hand, there are some much more tolerant microbes, things like Staphylococcus aureus, that has a very wide range of, hey, I can make it from 6 degrees Celsius. You can throw me in the fridge, like, hey, we're good, up to 46 degrees, much higher than even if the human body was running a temperature. Okay. And I have a little picture for your viewing pleasure over here. This is a ice sculpture from the area in Idaho that I'm from. And you might think that, hey, if you're making ice sculptures in McCall, Idaho, there's not going to be any microbes. And there might not be, but there are microbes that their cardinal range is very, very cold, and they could even be found in this block of ice. So there are 5 main categories of microbes when we talk about what range they prefer. So we can group them into psychrophiles, psychrotolerant philes, mesophiles, thermophiles, extreme thermophiles. And each of those are shown with these colorful curves. Okay, so the little dots show the minimum and maximum that we talked about a couple slides ago. And then the top of the curve shows their optimum range. So if we take one, for example, the green here, this would be where you would typically find human pathogenic microbes, or maybe even some of the good bacteria in your gut. Okay, their minimum range here is 10 degrees Celsius, their maximum about 50. And then the optimum range is about 37 degrees, which is our own body temperature. And you can do this for each of them. You'll see there are microbes surviving down to -20 below zero, so below freezing, and some that are surviving way past boiling being 100 degrees Celsius here. I would expect that the ones on the ends, the light blue curve and the kind of brownish curves, would be primarily in nature and would be prokaryotes, but they might be more archaea than bacteria. So I would expect on the cold end, you might find some weird archaea in the polar ice caps, and on the brown curve, you might find some interesting bacteria or archaea living in like thermal vents at Yellowstone or things like that. Okay, so some examples here of psychrochромophiles that are microbes with an optimum temperature below 15 degrees Celsius, typically around freezing, are going to be primarily in nature, as I was saying. And one example here is this red pigmented algae. You can see here, it's not like there's just dirt or something in the polar ice cap here. This is actually a living organism, an algae that's making this red pigment on the snow and ice. The psychrotolerant are going to be less extreme than the previous example, but they do tolerate cold. One interesting human pathogen is Listeria. So occasionally you'll hear stories on the news where there's been a Listeria outbreak, and it's typically associated with food going bad. And Listeria is unique because it can survive at cold temperatures in foods that are typically refrigerated or frozen. So you can even have Listeria outbreaks in ice cream, which is a crime in my opinion. Ice cream should not be, it should be off the table. No touching my ice cream, okay? But Listeria can grow in refrigerated meats and ice creams. Mesophiles are going to be the intermediate microbes that are typically the human associated organisms, whether they're pathogenic or not. They're going to be associated with them or other warm-blooded creatures because they grow between 20 degrees and 40 degrees Celsius. Thermophiles are going to be the heat-loving, greater than 45 degrees Celsius, but not too extreme yet. And then we still have our very extreme thermophiles. In addition to temperature, gases are also very important in the ecological role of microbial growth. The two main gases that often are discussed are oxygen and carbon dioxide. These are the ones that are most going to influence microbial growth, and that's partly because they're found all around us in the Earth's atmosphere. Microbes fall into three main categories: those that can use oxygen and can detoxify it, they aren't harmed by it, they have the right enzymes to kind of break it down. They can not use oxygen, nor can they detoxify it, which means it's not helpful, we don't need it, and it's actually hurtful because we can't detoxify it. And then there's some that don't use oxygen. It's not a necessary ingredient for them, really, but they can detoxify or break it down into usable substances. So this is an example of what you might see in the lab if you were growing a nutrient broth of different microbes. Okay, so you'll notice the cloudiness differs between each of these tubes, and that's because each of them has a different tolerance for oxygen. So in our first example up here on number 1, this one has to have oxygen. That's why you only see this kind of film of growth on the top and nothing down in the bottom. Okay. Number 2, it needs oxygen, but not quite as badly as number 1. So you'll see there's growth on the top and it grows into the solution here, and then there's a point at which it stops. 3 doesn't care. It can be at the top, at the bottom, anywhere. And 4 is actually trying to avoid the oxygen. So you'll see that it's much more cloudy at the bottom than it is at the top, which is closer to the oxygen. Microbes need to process the oxygen, and oxygen can only be acted on by enzymes. Now, oxygen by itself, there's singlet oxygen that can be toxic to the cell. And sometimes this is produced by phagocytic cells, like our white blood cells, to actually kill invasive bacteria. So it's useful in that respect, but it's not useful to a lot of microbes. So this oxygen is toxic and therefore needs to be dealt with. Hydrogen peroxide, superoxide ions, hydroxyl radicals, all can be damaging to the cell. And some of these just come from gases in the environment. They can also come from breaking down water, things like that. They're all around the cells and they have to find a way to deal with them. So cells have developed, have enzymes to help neutralize these chemicals. Okay, so some examples down here would be if you have this oxygen and hydrogen here, it can get broken down into hydrogen peroxide and oxygen, and then hydrogen peroxide can further be broken down by a catalase enzyme into water, which is not going to be harmful. It's actually going to be helpful for the cell. It's important to note that these enzymes just expedite the reaction. This reaction would normally happen on its own. It just would be very, very slow. So if I were to buy, those big brown bottles of hydrogen peroxide they sell at the drugstore, and I cracked the lid, opened it once, and put it back in the cupboard for a year, it would slowly degrade into water and the oxygen would be released. Okay, so that's going to happen on its own. Catalase is just helping to expedite this process. All microbes require some carbon dioxide somewhere in their metabolism. And we'll get more into metabolic pathways in our next chapter, but it's required for many different pathways. There are some microbes that will grow better in CO2 rich environments, some that grow better in oxygen rich environments. One of the most well-known microbes that does well in CO2 is Neisseria gonorrhea. Okay, so you can kind of look at, okay, where would I find this microbe? What would its environment look like? Okay, so Neisseria gonorrhea, well-known STD, would do well in CO2 rich environments. Okay, so another chart from your book here goes through the examples of the tubes I showed you earlier, showing aerobes are going to be the ones that can use oxygen in their metabolism and they can process it. Oxygen is required for obligate aerobes, and so therefore they're going to have this ring of growth at the top of the tube. Microaerophiles are harmed by normal atmospheric concentrations of oxygen, but they need a little bit of it. So they're not going to be up at the top where they're going to get a ton of oxygen, but they're going to be pretty near where they can still access it. Facultative anaerobes don't require oxygen, but use it if it's present. So they're just kind of like, hey, who's going to give me the best deal, basically. So they'll just grow all throughout the tube. They can live without it, they can live with it, take it or leave it, basically. Anaerobes cannot deal with the oxygen well, and so they're going to be avoiding it. They would be growing at the bottom of the tube and the top would be clear. And the aerotolerant anaerobes don't utilize oxygen, but they can tolerate it. So this one would look very similar to the facultative anaerobes when you look at the tubes. Okay, so in addition to gases and temperature, we also care about pH. pH is how acidic or how alkaline the environment is. Okay, so you can see down here, it's a scale from 0 to 14. If you think back to Gen Chem, the lower the number, the more acidic it is. The higher it is, the more alkaline. A neutral pH would be about 7. Your stomach acid is going to be very low down here. Okay. So most organisms prefer it to be near the neutral range. Being outside whatever their normal range though is can be damaging, especially to proteins and enzymes. Changes in the pH change how the proteins get folded. And then if they're not folded right, then they can't function right. And basically all the machinery gets broken down. Okay, so we have acidophiles would be in this red area where they prefer low pH. Alkalinophiles are very high pH in the blue range. Osmotic pressure is also important for microbial growth. Most microbes prefer a hypotonic solution, an environment in which there's more solute or more salts or sugars or things like that inside the cell, or an isotonic solution where it's equal. They would be trying to avoid the hypertonic solution where it would end up shriveling up. Okay. Osmophiles are microbes that can live in high salt environments, high solute environments, and prefer the hypertonic solution. These are often referred to as halophiles because they prefer salt. And we'll often find these in the environment, so places like salty lakes or salt marshes or ponds, things like that, like the halobacterium that inhabits salty lakes with a very high salt concentration of 25% sodium chloride or salt. Radiation or light is also a key attribute for microbial growth. Phototrophs, again, are organisms that use sunlight or visible light as an energy source. Most non-photosynthetic microbes, though, are actually damaged by the oxygen byproducts produced by contacting light. So we can get sunburned. The little microbes, all the bacteria need to put their sunscreen on. I know. So some microbes can produce yellow carotenoid pigments that basically act as like the melanin in our skin that protects us from light. They have their own pathways to make these carotenoid pigments. Now, that is from the microbe side. From the researcher or medical professional side, we know that most microbes are damaged by UV light. And we can exploit this weakness to use UV light to kill unwanted microbes. So you may have seen on like Amazon that they have like little boxes for phones and keys and things, and you can put your iPhone and your dorm key in the box, close the lid, turn the UV light on, and kill all of the bacteria. Some medical facilities use UV light to sterilize rooms that they're concerned have pathogens in them. We'll often use UV light to sterilize the chemical hood if I'm going to do cell culture, things like that. UV light kills the bacteria through damaging their DNA, and it also causes toxic oxygen byproducts. So between the two of those, it can be very detrimental to the microbe. Pressure is also a key attribute to consider. Barophiles are microbes that can exist under high pressure. This can range from a few times our atmospheric pressure to over 1,000 times. So typically these are ones that you would see very deep in the ocean, because if you think like scuba diving and you get the bends because you went down to a higher atmospheric pressure and then you came back up. Okay, so that would be the same thing for the microbes. It's difficult for most to be able to survive at the bottom of the ocean, but there are some pretty cool. Okay, so our last section on parameters here is on strong partnerships, or the microbes that are around them. Symbiosis is two organisms living together in close partnership. So this could be in the broader scope of ecology of like two organisms, like a certain plant likes to live near a certain animal, or two animals are working together. It can be positive or it could be negative. And the members of this partnership, both organisms, are known as symbionts. There are three main types of symbiosis, some positive, some negative. Mutualism is a mutually beneficial relationship where both are working together. It's usually one party is getting the nutrients and sharing it with their friend. The other one is getting some sort of protection or things like that. One example of this mutualism would be our gut flora. So there are microbes in your digestive tract, and we get protection, a healthier immune system. They keep the pathogens at bay, and they get to harvest a small cut of our nutrients, basically. Commensalism is when one party receives benefits, the other one doesn't care. It's neither helped nor hurt. Parasitism would be the host organism is going to be providing the parasite microbe with nutrients and a safe space and things like that, but it's costing them. So this would be pathogens. So if you have a microbe that you take in and you get food poisoning, well, the microbe's happy about it, but you're not. It was at your expense. There are other associations that are not quite partnerships. So non-symbiotic organisms are going to be free living. The relationships are not required, but they can do them or they can leave them. They can go either way, basically. So if we happen to be next to each other, sure, we can work together, but we'll still survive without each other. Antagonism would be when free living species associate and when members of the community are competing. So an example of this would be one microbe is secreting a chemical substance to keep all of the other microbes at bay because they want all the food. Okay, so you see this a lot in fungal species. So a lot of mushrooms will secrete chemical substances to keep bacteria at bay. And these chemical substances we've harvested for our benefit, and they're now antibiotics. Synergism is a relationship when multiple free living organisms work together and they all benefit. I've heard it said that synergism is like we added up each of the individuals and we put them all together, and the math doesn't add up. Somehow they're worth more together than they are apart. Okay, so a little flow chart to put this together in a different way. Organisms are associating with each other. They can be symbiotic or non-symbiotic. Symbiotic is when it is required by one member at least. So they're not going to make it on their own. Mutualism is we both need each other and we both benefit. Commensalism, one partner benefits, the other one doesn't care. And parasitism, where the parasite is dependent on the host to survive and the host is harmed. Non-symbiotic, it's the relationship is not required for survival, but we can do it if it happens to work out. So synergism, members cooperate and share their nutrients. They work together. Or antagonism, some members are inhibited, some are destroyed, some do better, some don't. That's like the antibiotic example.